Hello and welcome to our podcast on plants and people.
Hi, I'm Riley Moreland.
Hi, I'm Ramneek Nidger.
Hi, I'm Mary Sarumi.
And I'm Alexis Vieira.
We will be exploring if tannins that are typically in teas and coffees and wine can be helpful in treating illnesses. Our illness of focus was looking into diabetes. Many people with diabetes struggle with controlling starch indigestion, so there has been a hope for natural compounds to be able to reduce the rate of starch digestion. For our research, we looked into a paper that discussed Tannins hypocystis, a species of parasitic plant in the Tannins family. This plant is found primarily around the Mediterranean Sea and is ant-pollinated and grows within the roots of its host. These parasitic plants' juice is now traditionally used for treating several illnesses such as intestinal problems, inflammations, tumors, and bleeding in Sardinia. Since this species is rich in hydrolysable tannins, which are often associated with inhibiting starch digestion, the study is used two types of hydroethanolic extracts, heat-assisted and ultrasound-assisted, to see if it would affect the starch tolerance in mice.
We can relate the properties of these tannins back to a topic we have discussed in class, the coevolution of useful traits in plants. The study gives a snapshot into how a plant evolved biochemical defenses, tannins, that target essential digestive enzymes. These defenses may have originally served to deter herbivores and other pathogens. Humans are studying these defenses to explore their possible therapeutic potential in managing diabetes. This study was done in vivo on mice, so the lack of clinical trials done on humans is a limitation. Human trials would be necessary to confirm efficacy and safety in humans. The study also found that maltose digestion was not inhibited by the extracts containing tannins, which indicates that the effects may be substrate-specific and not effective against all carbohydrate digestion pathways. The strongest effects in diabetic mice were only seen at very high doses, which could also be unsafe or impractical in humans.
The study focused on the investigations of the potential of tannin-rich extracts from the parasitic plants Tannins hypocystis to inhibit the activity of alpha amylase, a key enzyme responsible for the breakdown of starch in the digestive system. The research focused on tannins, which is a class of polyphenol compounds known for their various biological activities, as the potential inhibitors present in the Tannins hypocystis extracts. The study was done in vitro assays, meaning in laboratory, to assess the alpha amylase inhibitory activity of Tannins hypocystis. This allowed for a controlled environment to observe the direct interaction between the plant extracts and the enzyme. Dose-dependent relationships that indicate higher concentrations of Tannins hypocystis extracts led to a greater inhibition of alpha amylase activity. This is a common characteristic of the enzyme inhibitor. Findings of the studies would suggest that tannin-rich extracts of Tannins hypocystis have the potential to slow down the digestion of starch in vitro.
The study explored the natural plant extract as a potential means to inflate the rate of starch digestion. The study investigates the ability of compounds with tannin to inhibit alpha amylase as a primary enzyme for breaking down starch into absorbed sugar. The inhibition of alpha amylase is a mechanism for potential reduce the rapid rise in sugar level after consuming starchy food. This is a significant challenge for many individuals with diabetes.
In conclusion, tannic acid has digestive enzymes that can reduce humans' digestibility and absorption of nutrients. We found that the C. hypocystis was able to reduce starch digestion in gastrointestinal tract. This can be important because this phenomena can help decrease the hyperglycemic excursion that occurs after a starch-rich meal for pre-diabetic individuals, and even help people who struggle with caloric intake and people who have difficulties with overeating.